So my job this morning is to try to convince you that B cells have something to do with lupus, and that targeting B cells with some selectivity might be a good approach to treating patients with lupus. Now, with the lack of success of rituximab in lupus and the modest success of belimumab in lupus, my job is pretty much like what you heard from Luke O'Neill trying to convince you about how therapeutically advantageous blocking IL-1 was in RA and OA. But I'll try to do the best that I can, because I think that there still is an interesting approach that can be used in targeting B cells much more specifically than has been done in the past in this disease. So here's a slide that everybody has. I think we've seen one version of it already this morning. You'll see a lot more. And this talks about the pathogenesis of lupus. And I think what we've learned from the multiple genetic studies that have been carried out now is that this is clearly a gene with an important genetic background. At least 30 gene polymorphisms have been associated with lupus. We expect, when the results of the immunochip come out in the quite near future, that perhaps as many as 50 or even 100 genetic polymorphisms are associated with this disease. And it is of some interest that there are now beginning to be great clustering of autoimmune diseases based upon the genetic polymorphisms upon which they are associated. And, diseases like MS and lupus and sometimes RA are beginning to look more similar than dissimilar. So it's a kind of an interesting time. In addition, we've had a lot of information emerging about the role of epigenetic control of gene expression and its role in developing lupus. But clearly, the combination of genetics and epigenetics generating an individual who has a particular capacity to respond in an unusual way. And I think another thing that's important about lupus is that most of the genetic polymorphisms that have come out so far actually seem to be involved in genes that regulate the immune or inflammatory systems, and we haven't seen a great number of genes that might affect host response to tissue damage. The other important aspect of lupus is the female predominance, and clearly sex hormones play a very large role in enhancing immune responsiveness. We've begun to learn something about environmental triggers of this disease besides UV light. Certainly cigarette smoking is a risk factor for this disease, perhaps because of its adjuvant-like capabilities, as it is for rheumatoid arthritis, and perhaps infections with microorganisms that stimulate toll-like receptors might also contribute to this disease. In lupus, there is very little work on potential role of the microbiome in contributing to lupus, but we do know that in the animal models of lupus, when these animals are introduced into germ-free environments, there's very little impact on the manifestations of this disease, implying that many of these models may not have a dramatic effect on the microbiome. Nonetheless, all of these things conspire together to lead to an individual with an immune dysregulation, which eventually leads to autoantibody production and lupus. So we're beginning to fill in the blanks in these various boxes. Now, among the immune dysfunctions that are critical are those that involve B lymphocytes, and I don't know anything about B lymphocyte metabolomics, so I won't talk about that at all. I'll strictly speak about the function of these cells and some of the influences that control them. And when I first started this, B cells were known as the precursors of cells that produced antibody, but since then a great number of functions have been identified, including their capacity to present antigen in certain circumstances, their robust capacity to produce certain cytokines that can regulate the immune system, their ability to downregulate the immune system by producing IL-10, their so-called B-reg function, and also their capacity to actually organize the environment in which they reside by expressing cell-bound lymphotoxin beta. So these cells have a great number of functions, and abnormalities in any or all of them could contribute to an autoimmune disease. Now, what do we know? I mean, I'm a human B-cell biologist, and I find the mouse extremely interesting and occasionally informative. But what do we actually know about the role of B cells in human lupus? Well, first, I can summarize the information in the mouse. A very, very, very large number of murine studies that are relevant. And that the information comes down to the fact that a variety of genetic manipulations in the mouse, all of which tend to enhance B cell function by manipulating one pathway or another, expression of one molecule or another, in a variety of mice, lead to autoantibody production. So there's a clear relationship between B cell function and the likelihood of producing autoantibody. In the right genetic background, these manipulations can lead to actually a lupus-like disease. And it's interesting that these B cell modifications to date haven't led to any signs and symptoms of inflammatory arthritis. So it looks as though B cell function is really linked to autoantibody production and sometimes in the right genetic background, the likelihood of developing lupus. In humans, a role for B cells in lupus or any other autoimmune disease really needs to be proven that's showing that some sort of an intervention that specifically targets B cells alters disease manifestations. And we really can only do that with some of the highly targeted probes that we now have. In lupus, of course, a role for B cells has been demonstrated by clinical improvement with therapeutic B cell targeting with belimumab. But all of you in the audience, and I'm sure we'll hear later in this conference, know that the impact of belimumab, which I'll talk about a little toward the end of my remarks, really has given only modest improvement in patients with lupus, asking whether or not this is really such a central target. Despite all the studies that have been carried out and all the clinical trials, except for the production of autoantibodies, the contribution of enhanced B cell function to the pathogenesis of lupus really remains uncertain, and specific B cell subsets involved, as well as the functions of B cells in lupus, have really not been resolved, and there's an opportunity for a great deal of additional work. So here is the life cycle of a B cell and in humans, and it helps to think of this because understanding this gives us an opportunity to monitor B cells and to monitor abnormalities in patients, and perhaps also monitor the impact of some of our interventions. B cells start life in the bone marrow, where they go through a series of molecular changes, generating an immature B cell that leaves the bone marrow into the circulation, and where it further undergoes maturation into a naive B cell that can enter a B cell follicle in secondary lymphoid organs, in the lymph nodes, in the spleen, and it resides there waiting for something to happen. And the something that happens is the encounter with antigen. And the nature of the antigen really determines to a large extent, what that B cell does. If the B cell is a multivalent antigen that really induces very poor T cell help, the B cell can respond in a T-independent manner, and that response consists largely of a rapid generation of short-lived plasma cells that make some IgM briefly, and this can be very important host defense mechanism for certain infections, and then the short-lived plasma cells die. If the antigen is one that can stimulate robust T cell help, then a different form of response occurs. This is a sort of T cell dependent response, which requires co-stimulation with T cells and the impact of a large number of T cell cytokines. And this T cell dependent response is often called a germinal center response. That's because during a germinal center response, there's very, very rapid B cell expansion, and so rapid that early histologists thought that these were the sites where lymphocytes were actually generated. But in fact, they're actually the sites where lymphocytes are responding in a T-dependent manner to antigen. This consists of clonal expansion. During the clonal expansion, these B cells their immunoglobulin genes can undergo somatic hypermutation and class switch recombination and lead to the production of a variety of effector cells of T cell dependent responses. These are memory cells, which have the capacity to respond in a more robust way to a second encounter with antigen. Again, a population of short-lived plasma cells that can make antibody for a short period of time and die, and then a population of long-lived plasma cells which can leave the germinal center, migrate through the blood, and come to reside in niches in bone marrow and in the gut and other places where they make antibody for a long period of time with no further stimulation. So the point here is that this is a very highly organized set of differentiation and maturation events, and that many of these stages, cells traverse the blood and can therefore be assessed in the blood. But here are the effector cells that we see just to remind you of what we're talking about. Memory B cells, these are generated during T cell dependent responses only. They're antigen specific. They're specialized for heightened secondary responses to antigen. They're usually expressed post-switch heavily mutated immunoglobulin genes or B cell receptors. And importantly, and it's been shown clinically and it's been shown in the mouse, in general, these are resistant to standard immunosuppressive therapy. They tend to be more resistant to rituximab, and they tend to survive after rituximab therapy, and they're completely resistant to the effects of BAFF blockaders, such as belimumab. And this will become important, I think, as we go on. Immunoglobulin secreting cells can be divided into two large families, plasma cells and plasmablasts, and the nomenclature around this is a little bit uncertain, but I'll give you a general idea so you'll understand that there are some very important differences between these cells. Plasmablasts are short-lived, dividing cells that secrete very large amounts of antibody transiently. So they're basically designed to protect you from dying acutely from certain microorganisms. They make antibody within 3 days of stimulation in vivo, and then they die. They can be generated during T-independent responses or during some T-dependent responses, at least transiently. They're antigen-specific. The ones generated during a TI response secrete IgM. Those generated during a TD response, T-cell-dependent response, secrete IgG. And in general, the mutational characteristics of the cells depend upon whether it's generated during a T-independent response, which are unmutated, or a T-dependent response, which are heavily mutated. These are the cells that are sensitive to immunosuppressive agents. They still express CD20 in humans, and they can be deleted by rituximab, and they're very sensitive to BAFF blockade with belimumab or with other regions. These are to be contrasted with plasma cells, which are long-lived, non-dividing cells. In fact, these may live for your entire lifetime after they're generated. They reside in specific niches in bone marrow and in mucosal-associated lymphoid tissue, and they secrete large amounts of antibody continuously without antigenic stimulation. And this accounts for almost all of your serologic memory, your memory to all of the immunizations you had as a child, and to most of the infectious agents you encountered during your life. They're generated during T-dependent responses. They're antigen-specific. They secrete IgG and IgA that's heavily mutated, and these are resistant to antiproliferative agents. They're resistant to rituximab, and they're resistant to belimumab. So this is the family of effector cells in the B cell axis. And you can look at them by simple flow cytometry, and everybody in this room has a pattern that's in general like this. This is CD19 positive B cells looking at immunoglobulin on the surface, looking at this marker that's helpful to separate naive from memory cells. And basically, there's a big population of naive cells. There's a smaller population of immature cells. And then there are 3 populations of memory cells, here, here, and here. This is pre-switch memory, post-switch memory. And this is an interesting population that's recently left germinal centers. And then there are plasma cells. So that's pretty much what B cells look like in normals. Now, from what we know, most of lupus really occurs as a result of an abnormally expanded germinal center. There are abnormalities in all stages of the B cell's life, and many aspects of B cell homeostasis are abnormal. This is a situation where you're not looking for the abnormality because everything is abnormal. You're looking for the relevant abnormality because there are only some of these changes that seem to contribute to disease. And that seems to be what's going on in the germinal center, where there's enhanced stimulation, somatic hypermutation, the generation of autoantibodies, and then selection, and I'll get into that a little bit later. The net result is the generation of large numbers of autoreactive memory cells and large amounts of autoreactive antibody and plasma cells, which can be detected. And you can detect that again easily by flow cytometry. Here again is a patient with lupus, and you can see just by comparison, looking at the same populations, you can see a major abnormality is the alteration in all of the post-germinal center cells with changes in all of the memory populations, and especially striking one in this population of memory cells, and also a dramatic expansion in plasma cells. So all of the post-germinal center populations are abnormal in most patients with lupus, again, focusing on the fact that it's really the germinal center reactivity that's abnormal in lupus and something that we really ought to be targeting. Now here I put together a hypothetical slide which tries to tell you more about the germinal center, and again, marries to it some of the GWAS data that's available, the genetic association data, but you have to understand that this is all hypothetical because even though a lot of polymorphisms have been identified and these are frequently tagged to genes, there's very little data to actually prove what the function of that polymorphism is, whether it changes the structure or the function or the biology of the markers that it's associated with, and even whether or not the polymorphism that may occur in a regulatory region of this gene is actually regulating another gene. So we do know that about 80% of these polymorphisms occur in regulatory regions, but we don't really know the impact of those polymorphisms. That being said, of about 30 or so genes or polymorphisms that have been identified in lupus, it is quite stunning that about 25% of them are candidates to control B cell function. And here I've listed some of these, and there are a couple that have also been identified that could clearly control T cell function. So we probably have a situation here where there is intrinsic B cell abnormalities, intrinsic B cell hyperresponsiveness, possibly in some individuals increased T cell help. A naive B cell is stimulated, and that has a great tendency to induce germinal center reactions. In that, there's expansion, somatic hypermutations, class switch recombination, and then the other abnormality in lupus that's essential, and that is an abnormality, especially in clearance of apoptotic material, that has the purpose of placing apoptotic material containing autoantigens in a position on follicular dendritic cells where they can actually select autoreactive clones, the net result of which is autoantibody secreting plasma cells and autoantibody reactive memory cells that come out of the germinal centers. And I put it to you that once you get this, that's when you have lupus, and once you have those cells, unless you eradicate them, you will continue to have recrudescences of lupus, and those populations tend to be resistant to the standard therapies that we use. So here's another way to look at this. Again, we're focusing on T-cell dependent response. Here are some of the interactions that occur between T and B cells that serve to generate initially short-lived plasma cells, and then with more intense prolonged interactions in germinal centers, long-lived plasma cells. And I show you this initially to make a point that different autoantibodies appear to be coming out of short-lived plasma cells and out of long-lived plasma cells. And depending upon the disease that we're treating, that may involve different kinds of therapies in order to target these specific populations. So we know that anti-DNA antibodies, which can be depleted quite dramatically with antiproliferative agents, with glucocorticoids, to a certain extent with rituximab, probably a large fraction of them are coming from short-lived plasmablast-like cells. Whereas other autoantibodies that don't budge with therapy, anti-SM, anti-RMP, these antibodies seem to be coming from long-lived plasma cells, and therapy is affects them minimally, if at all. And I think that's important to keep this in mind as to what would be the expectations of different kinds of therapeutic interventions. Okay, so let's look at this model and let's use it a little bit in order to understand what kind of therapies are in development that would tend to focus not on removing, deleting, or altering the function of all B cells. That's a kind of Quentin Tarantino approach. You know, you shoot everybody in the room and you assume that one or two might be bad guys. Probably better to try to actually identify the bad guys, and I think the bad guys are driving germinal centers. So how do germinal centers work, and can we target them? Well, first there's a specific stage of helper T cells called a T-follicular helper cell that resides within B cell follicles, and this population is the population that provides initial help to B cells. It migrates after stimulation to the TB interface, and it's the cell that initiates TB collaboration and starts the whole process. Without this cell, basically, you can't initiate germinal center reactions. We know that this cell is driven, importantly, by an interaction involving a surface molecule on the activated T-follicular helper cell, ICOS, and its ligand on B cells and other cells, ICOS ligand. So here's the story of ICOS ligand. ICOS is also called inducible T-cell stimulator, ICOS. It's a member of the CD28, CTLA4 family, and it's expressed on activated T cells. The ligand is a member of the CD80-86 family, and it's expressed on B lineage cells, dendritic cells, macrophages, and endothelial cells. But it appears as though B cells play a critical role in expressing ICOS ligand because B cells are required for the maintenance of T follicular helper cells. This population is critically involved in the generation of T follicular helper cells because it's involved in the maximal upregulation of a transcription factor, BCL6, which is essential to generate T follicular helper cells. In the mouse, we know that absence or markedly, that the absence of ICOS leads to markedly decreased germinal center reactions and decreased levels of IgG and IgE following immunization. And we know in humans, loss of function mutations are associated with a subset of CVID, so immunodeficiency. So this is central to germinal center reactions, and we also know that overexpression of ICOS in the mouse results in lupus. So in terms of a target, this is a very good one because you're looking at the non-redundant pathway that's essential to generate the T helper subset that's involved in germinal center reactions. Okay, what about this next one? So here we have a chemokine and a chemokine receptor, CXCL13 and CXCL5. And this chemokine and chemokine receptor is critically involved in repositioning T cells and B cells so that they can come together at the TB interface and initiate germinal center reactions. And many chemokines are not redundant. There are many chemokines that interact with many receptors, but this is one that's a non-redundant interaction, whose major function is to coordinate the positioning of T cells and B cells in the initiation of germinal center response. So CXCR5 is a typical chemokine receptor. It's expressed on B cells, on central memory CD4 cells, and also on T-follicular helper cells. So limited expression. It specifically recognizes this one chemokine, CXCL13, which is produced by follicular dendritic cells and certain stromal cells. So the location here is quite specific. The interactions of this chemokine and its receptors controls colocalization of activated T-follicular helper cells and B cells in the initiation of germinal center responses. And in the absence of this interaction, basically, germinal center formation and T-dependent antibody responses don't occur. So this is a non-redundant step in germinal center activity that is, again, a very good target to try to block germinal center activity in a disease like lupus. Well, the next thing is that after they finally get positioned and the T-follicular helper cell is activated, the interaction occurs uniquely through an interaction by CD40 on the B cell and CD40 ligand on the activated T cell. And what are these? Well, CD40 is expressed by B cells, also by antigen-presenting cells and endothelial cells. It interacts with its ligand, which is expressed on activated T cells, but also on platelets. This interaction is essential for germinal center formation. In the absence of the ligand in mice or humans, there's no germinal center formation, there's no class switch recombination, there's no somatic mutation, and there's no generation of memory cells. And in humans, these kids have hyper-IgM syndrome, a severe form of immunodeficiency. So this is another essential step in generating germinal center reactions and an excellent target for a disease that's mediated by germinal center reactions. Okay, so after you have activation of the T-follicular helper cell, and it can get together with a B cell, and there's some cellular cooperation, a cytokine is produced by the T cell that's essential to drive the rest of the response, and that's IL-21. Now, IL-21 is a type 1 cytokine. You can see it here. It's a member of the IL-2 family, and it has the same sort of composite receptor with signaling through the common gamma chain. It's a type 1 cytokine produced by activated CD4 cells, also CD8 cells and NK cells. It's constitutively expressed by the T-follicular helper cells that I mentioned. Some people call them Th21 cells. And interestingly, the gene has been gene polymorphism has been associated with lupus. The 21 receptor is a composite receptor composed of the IL-21 specific receptor and the common gamma chain. This signals largely through STAT3. The receptor is expressed initially on lymphohematopoietic cells, but now it's also known to be on epithelial cells, keratinocytes, synovial fibroblasts, and what have you. And the gene, the IL-21 specific gene, is also a polymorphism, is also associated with lupus. Major actions of IL-21 are to promote NK cell and CD8 cell survival, promote the differentiation of Th17 cells, but also it is the major cytokine that drives the differentiation of B cells into plasma cells during germinal center reactions. IL-21 in lupus, and there's an excellent preclinical story here. As I mentioned, IL-21, its biology is appropriate. It's known to drive plasma cell differentiation in both mouse and man. Levels of IL-21 are markedly elevated in some mouse models of lupus, including the BSXB mouse and the Sandrock mouse, and also in lupus patients. IL-21, lupus is associated with polymorphisms in both the IL-21 gene and the IL-21 receptor gene, and blocking IL-21 is effective therapy in the MRL-LPR mouse, and breeding the BXSB mouse onto the IL-21 receptor knockout basically deletes all aspects of this of this gene, of this mouse, all activation of T and B cells, all downstream effects in a mouse which is characterized by multiple copies of TLR7. That's the BXSYA mouse. So there's very powerful preclinical evidence that IL-21 indeed could be very important. Now, these are all specific, and now we get to the next player here, which is BAFF or BLyS. And BAFF or BLyS plays a role basically in maintaining the survival of B cells. It doesn't stimulate activation, it doesn't stimulate differentiation, but it seems to provide a survival signal, and the survival signal is largely for early plasmablasts and also for naive cells. Whether or not it's redundant remains to be seen. There is certainly a second cytokine that can do most of what BLyS does. So here's the BAFF BLyS story, and you can see on your left, BLyS synthesizes a transmembrane molecule cleaved by an ubiquitous enzyme, and then it can interact on B cells with one of 3 different receptors. But there is a second family member, APRIL, also synthesized in a different way, and it can interact with 2 of the receptors and also with cellular proteoglycans. These receptors are expressed on different B cells, naive, memory, and plasma cells, and they have different functions. And unless I forget to tell you, it seems to be that each of these plays a role in the development of lupus in the mouse, because knocking out any one of these receptors in the mouse has no effect on the disease, whereas depriving the mouse of BLyS effectively reduces lupus in the mouse. We know what BLyS does because basically there've been all of the cytokine and all the receptors been knocked out. Fundamentally, BAFF is essential for the differentiation of naive B cells. No BAFF, no naive B cells. APRIL seems to be more involved in plasma cell differentiation and switching to IgA. However, you need both of these cytokines for optimal plasma cell survival. Importantly, B cell memory, and I'll show you that in the human studies, is not affected by BAFF or BLyS at all. And that may be one of the reasons the drug belimumab is not more effective. The BAFF receptor on naive cells basically controls their maturation. No BAFF receptor or no BAFF, no naive B cells. TACI and BCMA are much more complicated, and they're expressed on more mature cells, BCMA on early plasma cells, TACI on memory cells, and these seem to have a negative effect on TACI seems to have a negative effect on the survival of memory cells, and basically when you remove it, you get increased and sometimes increased activated memory cells. So this is more confusing. BCMA contributes to plasma cell survival. So a somewhat confusing system. But what do we know? We know in mouse, if you overexpress BAFF, you get a lupus-like disease, which is lethal within 3 or 4 weeks. So it's a very, very profound disease. And we know if you block these one or both of these cytokines, you can treat animal models of lupus. Well, what about, so what's the rationale for BLyS blockade in lupus? Again, the data from animal models. There are elevated levels of BAFF and BLyS and APRIL in subjects with lupus, and we also know that the elevated levels can correlate with DNA and titers and with disease activity. So what happens in people? These are published data from various of the trials usually carried out with belimumab. Does belimumab work in the human as you'd expect in the mouse? And from the data in the mouse, and in fact, when you treat patients with belimumab, these colored lines, you get B cell depletion. And you can see that that's probably about 60% over the time of this therapy. All of the depletion, or most of it, affects naive B cells. There's about a 75% decline in naive B cells. Activated B cells are decreased. But the important thing here is that there's no decrease in memory B cells. In fact, there's a rapid increase in memory B cells, and that persists throughout the time of treatment. And that's been seen with all BLyS blocking agents. The mechanism isn't clear, but it may be a problem in terms of having increased circulation or circulating memory B cells, some of which are probably autoimmune. Plasma cells also decrease by about, high dose, maybe 30%, you can see here, and low dose less. So there is definitely effect on early plasma cells with administration of this. And you can see that's mirrored by a modest decrease in serum Ig levels. And there's also a decrease in some autoantibodies, as you can see here, for example, with anti-DNA antibodies in the placebo group. You can see a decrease in anti-DNA antibodies, but no decrease in some of the other ones that are coming from plasma cells like R and P. So you're getting a differential effect as be predicted from the biology. Now the problem was that there were two replicate studies that showed significant, although moderate, benefit. And one can debate as to whether or not how moderate that benefit was. From a B cell biologist point of view, that clearly shows a role for B cells in lupus. But the question is that the best that we can do? And my view is that, BAFF basically is tonic. It doesn't get memory B cells. It basically functions to keep these cells alive. APRIL can do that as well, perhaps other things. And perhaps if we focus on some of these other targets that are much less redundant, we might get a much better effect in terms of treating lupus by treating germinal center reactions. The last one I'd like to talk about a little bit in the last couple of minutes is IL-6. IL-6 plays a critical role in the terminal differentiation of plasma cells through signaling through STAT3. And it also could contribute to lupus pathogenesis. There are a lot of indications from the older literature that IL-6 plays a big role in lupus. In other words, you can find increased serum levels in active lupus patients. You can find increased production by T and B cells in vitro, increased receptor expression. And if you culture lupus T and B cells in vitro, blocking IL-6 decreases the spontaneous production of Ig and anti-DNA antibodies. IL-6 is known to increase mesangial cell proliferation in the kidney. It's known that there's a lot of IL-6 in the kidney, and you can even monitor lupus patients by monitoring urinary IL-6 levels. So based upon that, at the NIH, we got quite interested in whether or not we could have an effect on lupus by blocking IL-6, and we used tocilizumab, which was available at that time. And this study is so old, it started when tocilizumab was being developed by the Chugai company, which was subsequently purchased by Roche. The idea was, this was a proof of concept study. We wanted to see whether blocking IL-6 had an impact on lupus. And so you can see what we did. It's a dose escalation study with a modest number of patients. We used very high doses of the study, and we were basically treating patients with modest disease with very low background levels of other drugs, and we were looking for some kind of clinical benefit and toxicity. So this is what happens in these lupus patients when you block IL-6, and basically there's a prompt decline in all acute phase reactants down into the normal levels, and this is also true of CRP and amyloid-associated protein. And as soon as you stop the drug, of course, the acute phase reactants come back. You can see by 14 weeks that there's a decline in anti-DNA antibody titers, which begin to increase after the drug is stopped. In terms of disease measures, SLEDAI, SLAM, BILAG, all of those things, you saw a significant decrease. In fact, at the levels that would have meant responder in the belimumab trials. And the most interesting thing for us was that if you looked in lupus patients, you could see this large population of circulating plasma cells. You can see a fair percentage of patients have increased plasma cells. And after treatment, you can see that the circulating plasma cells came back into the normal range. And you can see that for all the patients here. And in fact, all of the abnormal T and B cell subsets we could see also returned to normal, suggesting that IL-6 was playing a much greater role in abnormalities that we could measure in T and B cells than we had actually appreciated. So prompt decrease in acute phase reactants, decrease in anti-DNA antibodies, improvement in overall disease activity, and normalization of B and T cell subsets. This was an uncontrolled trial, and now there are 2 or 3 companies that are carrying out controlled trials of IL-6 interventions in some sort of lupus. So let me finish up by this. I think that by looking at the specific steps that are non-redundant in driving germinal center reactions and driving T-cell dependent B-cell responses, we may have a much greater effect in lupus than just doing non-specific depletion of the cells. And for example, with rituximab, depletion of germinal center B cells is quite modest. So by looking at these steps, thinking about them, and beginning to think about targeting T-follicular helper cells, IL-21, CD40 ligand, perhaps IL-6, one might get a much more selective and effective therapy of lupus. And I believe that there are companies that are developing products to target all of these non-redundant steps. So thank you very much for your attention.